For billions of years, life on Earth has evolved through slow, natural change, guided by the rules of DNA and natural selection. But now humanity has learned to read and edit this ancient script. We stand at the threshold of a new power, the gene drive. Imagine rewriting a sentence in the book of life, and watching that change spread through every new page, every generation. A gene drive doesn't just alter an individual, it ensures that change rapidly spreads through a population. This technology gives us the ability to decide which species thrive, which are modified, and which may disappear forever. It's a profound responsibility, once reserved for the slow forces of nature. The story of gene drives is a story about us, our ingenuity, our compassion, and our hubris. We dream of eradicating disease not just with medicine, but by editing the creatures that carry it. This power forces us to ask, when is it right to intervene so deeply in nature? Our journey began with Mendel's pea plants and has led to CRISPR, a tool that lets us edit genes with precision. Each step brought us closer to this moment, a technology that could reshape our world for the better. But with great power comes the need for even greater wisdom. The universe often reminds us that our understanding is never complete. For millennia, humanity has battled a tiny but deadly foe, the mosquito, not for its bite, but for the diseases it spreads, most notably, malaria. Malaria has shaped history, causing immense suffering and death, especially among children in sub-Saharan Africa. We've fought back with insecticides, bed nets, and medicines, but mosquitoes and parasites evolve, always staying a step ahead. Enter the gene drive. What if we could edit mosquitoes to make them incapable of transmitting malaria? Scientists have engineered mosquitoes with genes that block the parasite, but spreading these genes through wild populations was impossible, until now. A gene drive ensures that nearly all offspring inherit the beneficial trait, allowing it to spread rapidly. In models, releasing a small number of modified mosquitoes could transform entire populations in just a few years. We could create a world of malaria-proof mosquitoes, saving hundreds of thousands of lives annually. This would be a public health victory of historic proportions, all by rewriting a few lines of DNA. But such power demands careful consideration. The stakes are nothing less than the future of entire species, and the consequences, for better or worse, will be global. To grasp gene drives, let's revisit Mendel's laws. Normally, offspring have a 50-50 chance of inheriting a gene from each parent. This randomness ensures diversity and has shaped evolution for eons. A gene drive breaks these rules. It cheats at inheritance. Using tools like CRISPR, a gene drive copies itself onto both chromosomes, ensuring nearly all offspring inherit the change. The result? A trait that would normally fade away instead spreads exponentially. This is biased inheritance, a thumb on evolution's scale. We could use gene drives to spread malaria resistance in mosquitoes, or even engineer populations to produce only males, crashing their numbers. Extinction on demand becomes possible. Such power is unprecedented. It's a tool that commands change, not just suggests it. But with this comes immense risk. We must weigh the benefits against the potential for irreversible consequences. Gene drives could solve persistent problems, or create new ones. The choice is ours, but the responsibility is enormous. Hawaii, a paradise of unique species, is now a graveyard for many native birds. The culprit mosquitoes, introduced by humans, carrying avian malaria. Native birds, with no immunity, are confined to shrinking high-altitude refuges as the climate warms. Conservationists have tried everything, restoring forests, building fences, but these are temporary fixes. A gene drive offers a new hope, targeting the invasive mosquitoes, not the birds. Scientists are developing gene drives to either block the disease or crash mosquito populations. This is a shift in conservation, using genetic tools to remove a specific threat, not just build barriers. The goal, restore balance and give native birds a fighting chance. It's about healing nature, not conquering it. A genetic arc for Hawaii's birds, carrying them into the future. But even with the best intentions, the risks must be carefully weighed. The stakes are the survival of an entire ecosystem. New Zealand, shaped by isolation, is home to unique flightless birds like the kiwi. The arrival of humans and invasive mammals brought devastation. Billions are spent fighting rats, stoats, and possums, but extinction still looms. The nation's bold goal: predator-free 2050, eradicate all invasive mammals by mid-century. Traditional methods alone may not be enough. Gene drives could be the breakthrough, releasing modified rats with a daughterless gene drive to collapse populations. The same could apply to stoats and possums. This tool could achieve in years what would take centuries otherwise, with less collateral damage. But New Zealand is cautious, asking hard questions about risk, ethics, and consent. They could be the first to use gene drives for targeted extinction. The world is watching. The outcome will set a precedent for conservation everywhere. The balance between ambition and caution is critical. Gene drives are powerful, but their risks are just as great. The biggest fear: escape. A gene drive released in one region could spread worldwide, beyond our control. There's also the risk of the drive jumping to non-target species, with unpredictable ecological consequences. Ecosystems are complex. A single change can unravel the whole web. Nature fights back too. Organisms could evolve resistance, rendering the drive ineffective or even creating new problems. These risks aren't just theoretical. They're real challenges scientists are racing to address. We're manipulating evolution itself, venturing into uncharted territory. Small genetic changes can have massive, cascading effects. Humility and caution are essential. Once released, a gene drive can't easily be recalled. The genie, once out, may never go back in the bottle. We must proceed with care, or risk consequences we can't foresee. Every powerful technology has a dark side. Gene drives could save lives, but in the wrong hands, they could be weaponized. Imagine a gene drive targeting pollinators like honeybees, triggering ecological collapse and famine. These concerns are real, not science fiction. Agencies, like DARPA, fund gene drive research to develop countermeasures, not just cures. Reversal and immunizing drives are being explored to defend against rogue releases. This is a new kind of arms race, one fought in the code of life. A gene drive could be developed in a small lab and released with global consequences. The international community is only beginning to grapple with these threats. Scientists find themselves developing tools for both healing and harm. Once the knowledge is out, it can't be unlearned. Our wisdom must keep pace with our science. Gene drives don't respect borders. A modified organism can travel and spread globally. This raises unprecedented governance challenges. Who decides to release a technology that could alter a species worldwide? Countries suffering from disease may want gene drives, while others fear ecological risks. There's no global authority to make these decisions for all humanity. The decision requires broad, inclusive consent, especially from affected communities. It can't be imposed from afar. Trust and transparency are essential. We stand at a crossroads: coordinated, cautious action, or chaotic, unilateral releases. The technology forces us to think as one species on a shared planet. Our divisions seem small compared to the power to rewrite life itself. We need a new kind of global conversation, urgently. The process matters as much as the outcome. The future of our world depends on how we choose to act together. Fears of runaway gene drives have inspired safer designs. The daisy chain gene drive is one such innovation, powerful, but with built-in limits. Instead of a single, unstoppable element, it's a series of genetic links that depend on each other. As generations pass, the links break, and the drive fizzles out. This self-limiting approach allows for local, targeted changes without global risk. The drive can do its job, then fade away, leaving only the desired trait. It's a middle ground, powerful but restrained. A daisy chain drive could eliminate invasive mosquitoes from an island without threatening the planet. Built-in safety switches make the technology more acceptable and reduce catastrophic risks. It's a sign of growing maturity, engineering not just power, but restraint. The question is not just what we can do, but what we choose not to do. The future of gene drives will be shaped by our wisdom as much as our science,